Hello and welcome to this video. In today's video we're going to be discussing chloroplasts, pigments found within them, and the scientists involved with furthering the knowledge of photosynthesis as well as plants. So, by the time this is all said and done, we should have a good handling on what is needed for photosynthesis as well as the history of how the process was discovered. So, let's go ahead and get started. So let's go ahead and look at the internal structure of a chloroplast. So a chloroplast in plant cells is the structure where photosynthesis will be taking place, as well as its two major reactions. So the internal structure of a chloroplast we're going to be comparing to breakfast. Now I know that's a little bit out there, but trust me, it might go ahead and help you remember it. So the first thing we need to discuss is a thylakoid. A thylakoid is an internal membrane and looks similar to a pancake. So these are going to be tiny little green discs inside the chloroplast. Now these are often found in stacks, like a stack of pancakes. Now, the thylakoid. This is where one of the key components and reactions of photosynthesis is going to be taking place. Up next is called the lumen. The lumen is the space found within the thylakoid. So if we're comparing a thylakoid to a pancake, this would be like the light, fluffy structure inside the pancake. Up next is we have a grana. A grana is what is referred to as a stack of thylakoids. So if you see a stack of thylakoids, we can also determine that to be a grana. So think of it like this. A grana is a stack of pancakes. If you really want to go ahead and confuse somebody, the next time you go to IHOP, ask for a grana of pancakes. They'll have no idea what it is you're talking about. Up next, and finally, we have the stroma. The stroma is the dark, viscous liquid that the thylakoids are embedded in. Now, I don't know about you, but when I think of a dark liquid that's often put on pancakes, I often think of syrup. So the stroma is like the syrup that goes on the pancakes. So the stroma is the solution that the thylakoids are found in. It's kind of like cytoplasm for the chloroplast. Now, with this is where the second of the two reactions of photosynthesis is going to take place. But we'll get to that point in the next video. So, to go ahead and run through this one more time. Like we said, we have the thylakoid, which is corresponding to one pancake. We have the lumen, which would be the area inside of the thylakoid. We have the grana, or granum, which is the stack of pancakes. And then inside all of this, we have the stroma, which is the liquid inside of the chloroplast that surrounds all of our thylakoids. There you have it. Up next, we need to go ahead and discuss pigment. Now what pigment is are light-absorbing molecules. Each pigment absorbs and reflects different wavelengths of light. And the wavelength that we see, the color that we go ahead and perceive when we look at something, is the wavelength that is reflected. So if you take a look, we have a wide variety of different types of wavelengths. We have gamma rays, X-rays, UV rays, infrared rays, radar, FM, TV, shortwave, and AM radio waves. Now, there is a very small spectrum right here, ranging from a little under 400 to above 700 nanometers for wavelengths, this area right here, that is referred to as visible light. So, certain pigments are going to go ahead and absorb certain wavelengths of light, while they go ahead and can't absorb and reflect others. And the color that we see is whatever the wavelength is reflected or not absorbed. So, in plants, specifically photosynthetic ones, well, most plants in general, that was kind of redundant. Chlorophyll is the primary pigment in photosynthesis, and this is what goes ahead and gives plants their green color. So if you think back to it, this means that plants actually cannot absorb the green wavelength of light, and it's reflected, hence why plants appear green. Now, there are other pigments inside of a chlorophyll as well, a chloroplast as well. These are called carotenoids. They are accessory pigments that absorb light that the chlorophyll cannot. And these are primarily yellow, orange, and brown. So if you think about when the temperature starts to get colder and there's not as much sunlight, so think autumn. Trees go ahead and their leaves gradually turn from green to brown, yellow, and orange. However, these accessory pigments can't go ahead and efficiently work as well as the chlorophyll. So what happens is they cannot support the lifespan of the leaf, can't give it enough energy, and hence why the leaves go ahead and will fall from the trees and give us those wonderful colors we see in autumn. the more you know. This is just a real nice visual representation of what we see here regarding pigmentation and color. So the sun gives off a whitish light, which is the mixture of all of the different colors of the spectrum. However, in this case, with this pigment right here, it cannot absorb the green wavelength. So it's absorbing red, blue, and every other wavelength, but reflecting green. So the color that we perceive that pigment to be is the one that's reflected. So the green pigmentation. Finally, we're going to go ahead and discuss 3 scientists that went ahead and lent some knowledge into plants and how they work. The first one is von Helmont. So von Helmont measured plant mass and soil mass before and after 5 years of growth. And he found that most of a plant's gain in mass did not come from the soil, but came from water. So if you take a look at his experiment here, he took a 5-pound willow sapling and 200 pounds of dirt, put it in a pot. After that, he let the plant grow for 5 years, and after 5 years, the plant went from 5 pounds to 189 pounds and 3 ounces, which was a 3,400% increase in weight. Meanwhile, the dirt weighed only 199 pounds and 14 ounces. So, if the plant wasn't going ahead and getting its mass from the dirt, it had to be getting it from somewhere, and the only other thing he used in the plant was water. So he determined that plants get all their mass from water, which makes sense considering they have very large vacuoles used for storing the stuff. Up next is Priestley. So Priestley went ahead and took a candle, placed it in a jar, and then would cover the jar. And sure enough, he found that the candle would die out over time, because remember, fire needs oxygen to burn. However, the next time he did it, he went ahead and put a tiny piece of plant, so a tiny little sprig or bit of plant matter, inside the jar as well as the candle. And sure enough, he found that the candle burned for a longer period of time. So he deduced that plants must be producing oxygen, because the candle burned for a longer period of time, and like we said, fire needs oxygen to burn. The final scientist we're going to discuss is Ingenhousz. So Ingenhousz went ahead and did the same experiment that Priestley did, except this time he went ahead and did one in the light and one in the darkness. So, the setup that he had during the light went ahead and caused the candle to burn for a longer period of time, whereas he had another jar and candle setup that he placed in a dark room. And sure enough, the candle didn't really last as long. So he determined, okay, well, if the plant went ahead and was producing oxygen, allowing the candle to burn for a longer period of time in the light, but not in the dark, that must mean that the only time you can get oxygen from a plant is when it is light. So with that, that's going to go ahead and close up shop here on what you're going to need to know going into photosynthesis. If you have any questions, please go ahead and contact your instructor and they'll point you in the right direction. So with that out of the way, thank you very much for your time, and until the next video, take care of yourself.